Welcome, everyone. Welcome, everyone. Good afternoon. It's good to see all of you. It's a pleasure today to introduce Joe Loscalzo to you. Joe is a visiting professor from the Brigham and Women's Hospital from Harvard Medical Center School, and Joe is the Hersey Professor of Theory and Practice of Medicine at the Brigham and Women's Medical Center. Joe received his bachelor's degree as well as his MD degree from the University of Pennsylvania, where he also received a PhD in biochemistry. He, during the course of his studies, was elected to Phi Beta Kappa as well as Alpha Omega Alpha. He went on to a residency as well as chief residency and fellowship at the Brigham and Women's Hospital, where he focused on cardiology. He has served as chair of medicine at Boston University, and for the past 15 years has been chair of medicine at the Brigham and Women's Hospital. Now, you can say this about very few people, that they've established a field, but Joe has really established this field of network medicine as it relates to the biology, physiology, and environmental factors that are related to the development and progression of many of the diseases that we take care of. And this approach has resulted in the novel understanding that many of the diseases that we take care of are related biologically, physiologically, and even therapeutically to each other, and are not distinct diseases but represent spectrum of disease activities. Joe has been recognized nationally, internationally for this contribution. He's been elected to American Society for Clinical Investigation, to the Association of American Physicians, to the National Academy of Medicine. Please join me in welcoming Joe Loscalzo.
Thank you. Thanks very much, David, for that very kind introduction. I'm happy to visit again and meet many old friends and some new ones, and talk with you today a little bit about this notion of network medicine, what it means, how it may influence our discussions about disease causation and treatment, and how it may, as a consequence, have an effect on precision medicine as that field evolves. So the outline of the presentation is shown here. There are 4 parts to it. One is to talk about the problem of disease definition as we define disease currently, then touch on genomics and disease and new ways to think about applying genomic data to disease understanding and molecular basis. Talk about phenomics and precision medicine, how we characterize the phenotype of disease, and then talk about therapeutic implications. So I'll begin with 2 questions. Why has the rapidly expanding knowledge of the human genome not yet given us the expected epiphanies about complex human disease? And then why has new drug development been limited despite a remarkable range of technologies that are now used to identify drug targets rather specifically and precisely and design pharmacologically suitable candidate drugs? And obviously, since I present these 2 questions on the same slide, I believe the answer is a common answer, common to both of them. And it has to do with a changing scientific paradigm that we have all been taught from junior high school about Cartesian reductionism, which stems from the Greek philosopher Democritus, who talked about atavism, that we should explore systems when pursuing inquiry about how a system works, using what has become the conventional scientific approach: hold all variables constant except the one of interest, and then deduce its importance, inductively generalize, and then draw conclusions that may be relevant in the case of our field of biomedicine to treatment or to a disease course. And while this reductionist approach has been extraordinarily helpful over the years, not just in our sciences, but also in the physical sciences, it often oversimplifies complex systems, among which are included the complex biological systems that underpin complex phenotypes. And it leads to linear thinking, about which I'll say much more. And a timely example that I'll use, I'll set the stage for by giving you a case presentation. This is a case of a 39-year-old man who presents with new onset painful left calf swelling. He had no prior medical history. There was no history of trauma to the leg, no recent surgery, nor any recent lengthy travel period or period of bed rest. He had no significant past medical history remotely either, and took no medications, had no allergies. And on examination, he was tall, thin, resting comfortably. He was afebrile. He was not tachycardic. His blood pressure was normal, and he had an O2 saturation of 96% on room air. He had downward dislocation of the left lens and a high arched palate with a chest that was clear to percussion and auscultation. His jugular venous pressure was 9 centimeters of water at 45 degrees. He had no increased P2 and no murmurs or rubs. And the only other notable finding was that his left leg was swollen at the calf and distal thigh, and he had a positive Homans sign. And here's a picture of the, at least, global phenotype of this young man. He was tall, thin. He has a valgus deformity of his knees, as you can see. You can also appreciate that he has a pectus deformity as well as long, spidery fingers. And he had dolichostenomelia, not illustrated here, where his arm span was greater than his height. His initial laboratory tests were normal except for the D-dimer of 3540. He had no evidence of a PE by CTPA. His ECG was normal, but his non-invasive lower extremity study showed a DVT of the left proximal femoral vein. So in addition to initiating anticoagulation with heparin, what studies would you perform next in this spontaneous DVT, in an otherwise young man with this body type? Well, of course, he had a factor V Leiden and a prothrombin gene mutation tested, but also a homocysteine level measured, which was significantly elevated, and that's illustrated in the pedigree here. His homocysteine concentration was 389 micromolar, which is about 20-fold higher than normal. So this leads to a couple of comments about the homocysteine theory of atherothrombosis. And I think it'll become clear in a few moments as to why I'm using this disease as a way to illustrate the changing scientific paradigm, why it's necessary and essential for our future explorations of disease and treatment. This theory was first proposed by the pathologist McCulley, who, after performing autopsies on young individuals who died with homocystinuria, a rare inborn error of metabolism, in which they developed premature atherosclerosis or arteriosclerosis in the second or third decades of life. He argued that, in fact, even mild degrees of elevated homocysteine may be an independent risk factor for atherothrombosis. And over the next 30 years, many studies, epidemiological studies, supported that association, that homocysteine was a significant independent risk for atherothrombosis. And it is found in these mild degrees, say in the 14 to 20 micromolar range, in up to 40% of patients with defined vascular atherothrombotic disease, but in only 2% of unaffected individuals. So based on that epidemiology, the initial observations in children with the rare inborn error of metabolism, and knowledge of homocysteine metabolism, which is summarized here. Homocysteine, remember, sits between the remethylation pathway that takes us back to methionine and takes homocysteine back to methionine in the transsulfuration pathway. And notice also that there are a variety of B vitamins that are crucial for these metabolic conversions. B6, folate and B12, betaine, which is trimethylglycine. So based on this knowledge of the relationship of homocysteine to atherothrombotic disease and the clear B vitamin dependence of its metabolism, there were 7 clinical trials which were established in the late 90s. Those continued through the mid-2000s, early part of this century, the first decade of this century. And all of them yielded the same result. These were all trials of patients for secondary prevention. They had established atherothrombotic disease or its diabetes equivalent. And what was observed was that none of these patients had any benefit from these treatments. The combinations of B vitamins were different trial to trial. The entry criteria were different, but the outcomes were virtually all the same, as illustrated here by the Norvid and HOPE-2 trial, the first 2 of the trials that were published in the New England Journal. So following these results, there was lots of pulling of hair and gnashing of teeth, and the question that was raised by the clinical trial community involved in these studies was, how could we have been so wrong? Here we have homocysteine. It can easily be lowered by folate or B12 supplementation in most individuals, or by B6 supplementation. And yet there was no benefit with respect to clear clinical endpoints. Well, I would say we should turn this question on its head and ask, how could we have been so naive to think there should have been an effect? And the reason I say that is illustrated in this slide. We begin with a small element of a larger metabolic map that highlights where homocysteine sits relevant or relative to folate metabolism, which of course is contained within a much more complicated set of pathways that govern folate and B12 metabolism for many other important cell functions, which of course sits within an incredibly complicated metabolic map. So you could ask the question, how could we have been so foolish to believe that by simply tweaking this system with a little additional folate or B12, we could have that much of a benefit for this complicated pathophenotype? And of course, in retrospect, it became painfully obvious as to why there might not have been a benefit. Folate and B12 have many other effects which could adversely affect the consequences of established atherosclerosis, not the least of which is their effects on DNA synthesis and cell proliferation. There are also effects on methylation because homocysteine sits in an important remethylation cycle. The relationship between homocysteine and the SAM-SAH ratio, which governs all methylation pathways or reactions in the body, the specificity being driven by the specific methyltransferase. That ratio is altered dramatically by elevated homocysteine, which has adverse effects for epigenesis, for nitric oxide production, and for many other features of normal vascular biology that could have contributed to the poor outcome. So I use this case as an illustration, and those of you in other subspecialties, as well as in cardiology, in other areas of cardiology, can no doubt recount many clinical trials that have yielded very similar negative results in your fields, despite an abundance of clear reductionist data in cell culture and animal model systems that support a particular therapeutic strategy. And I think one way to look at these negative trials in a constructive and proactive way is to recognize that most biological systems can't be reduced in practice when tested in clinical trials to what one can do in tissue culture. These are systems that respond to multiple inputs that vary simultaneously and can interact. By definition, these are complex systems that form molecular networks, and we'll talk more about that in a moment. And what I'll try to show you in the next few slides is that there are new quantitative approaches that can be used to construct these networks, identify their dynamics, how they may contribute to disease pathobiology, and examine their responses to perturbations, including drug-induced perturbations that may restore normal function. So just a couple of slides on networks and network science and how that can apply to biological systems. Most networks that one can explore in biological systems are not stochastic. That is, these proteins don't associate or these genes don't associate in a purely random way. They are frequently, if not invariably, clustered or scale-free, and I'll tell you what I mean by that in a moment. They also manifest emergent behavior, which means that if you were to study one element of that system as rigorously as you can, know everything about that gene, everything about its regulation, everything about its dynamic expression, that despite knowing everything you can about that one element in the network, you cannot predict from a knowledge of that one element how the network itself will behave. Sort of like trying to predict how an electrical network would behave if all you knew were the principal features of one resistor in the network. And I would say, well, that seems like a trivial analogy. It is the way we do our science, both at the basic level and at the clinical level. We focus on that one enzyme or that one gene or that one resistor. The emergent behaviors are then an afterthought and lead to our surprise when we notice that the outcome wasn't as predicted. So another important quality of these biological networks is that functional modularity really follows structural localization. So there are networks that sit within different compartments of the cells, and they communicate with each other through weak links between clustered networks. The reason this is important, this clustering is important, one reason it's important, is that it minimizes the time it takes for information to flow across the network. Consider, if you will, the difference between a high school class in which there are cliques of people, you know, the geeks and the prom queens and kings. And they sort of they create their own little social networks, and once in a while there'll be somebody who serves as a weak link between those cliques. And if you want information to flow across the whole high school class, you have to know where the weak links are. That's how gossip travels across the high school class. The same principle applies here, that these clustered networks really facilitate the transition time between states. They make the entire network more efficient. And they comprise canonical, structural, and dynamic motifs that really are the basic organizing principles of systems biology, much like the genetic code is for DNA. So here we see an example of two networks. On the left, a random network where the nodes are linked randomly one to another by links or edges. On the right, we have a scale-free or clustered network. And here you can see that most of the nodes have single links to the network, but there are three nodes, shown in blue, which are called hubs, that are enriched and have many more links. And as you can see, the random network is defined by a Poisson distribution, where K are the nodal connections or links, whereas the scale-free network in a log PK plot is defined by a slope, the gamma, which is invariant with respect to the size of the network. And notice that this graph really indicates that there are many nodes that are sparsely linked, the peripheral nodes, and there are only a very few nodes that are heavily linked. These are the hubs. And this invariance with respect to size defines these networks as scale-free. And that's important in biological systems because this is the way biological systems grow. That is, the rich nodes get richer. The rich genes or gene products are more likely to link to newly adaptive gene products in the course of evolution. So these scale-free networks have biological implications that include this notion of natural selection and evolution. They define the difference between mutable nodes, which are weakly connected. These are the peripheral nodes that engender diversity, biological diversity, and facilitate natural selection. And as I'll say more of in a moment, are also the nodes that are responsible for complex chronic diseases. But then there are also the immutable nodes. These are the hubs, the loss of which can be embryonically lethal, such as G6PD. So the other important feature of a scale-free network for biology is that it accommodates perturbations to the network with minimal effects on critical functions of the organism, unless the hubs are perturbed. So it decreases the frequency, assuming that an external perturbation occurs randomly, decreases the likelihood that a network will go down, that its behavior will be arrested as a consequence of a perturbation that occurs to any one of the elements of the network. So now I want to digress for a moment and talk about linear thinking from a different perspective, and begin with this slide, which shows a typical Manhattan plot. This is taken from the Wellcome Trust Case Consortium, one of the first major disease GWAS studies. And it highlights one feature that I want to emphasize here, and that is the linear display of the genome. And I'll make the case here that we're used to communicating in a linear way. You know, aside from members of the legal profession who can speak out of both sides of their mouth simultaneously, the rest of us don't have that skill. And we're forced to share knowledge, ideas, in a very rigorous kind of sequential way, ad seriatim, building an argument. And that means of communication has been translated into many other features of our normal working lives, including as biomedical scientists, the way we think about the genome. We think about it as a linear structure as depicted here. And I would say that there are many shortcomings of, while it's important and useful and certainly simplifies the problem of linking genes to disease, it oversimplifies the problem and really does bury the problem by not uncovering the complexity that exists among the relationships in those loci. It would be as if you, as an engineer, were given a list of parts and asked to reconstruct the engine that these parts comprise elements of. And in your efforts to try to sort out why the engine isn't running properly, or maybe there's a little chip in one of these gears here, and you're assuming that that's what's causing the problem, but you really don't know what the assembly diagram is. You just know that that gear is different from one in another car that looks the same, whose parts you've been shown. And what you need, of course, is the wiring diagram. You can't really understand how the parts work together or how that variant, that chipped cog in that gear, might make a difference with respect to function if you don't know the diagram. And I would say, without pushing this analogy too far, that the best way to express it is, as Rutherford did, that all of science is either physics or stamp collecting. And while collecting genetic information the way we have had to do so over the last 15 to 20 years is absolutely essential, it's time for the physicists to enter the picture and begin to assemble them into meaningful networks. So that leads to at least one definition of network medicine, as I'll use today. It's the study of disease as viewed through the prism of networks of interacting mediators. These can be genes, proteins, metabolites, physiologic mediators, and their responses to perturbation, which can be a naturally occurring pathological variant. It could be a therapeutic perturbation. It could be an environmental exposure. And the question we need to ask is, can molecular networks then give any unique insights into disease pathogenesis and therapy? How much better is it than just looking for these simple one-off associations and pursuing them to their ultimate reductionist expression in some cell or animal model? Well, to answer the question, let me first say that I think the answer to the question is yes. Otherwise, I'd be giving a different talk. I'll try to make the next few slides. This slide, taken from a review that Laszlo Barabasi and I wrote a few years ago, makes the case that disease genes, at least in chronic complex adult diseases, not strict Mendelian diseases in early life, are largely non-essential and do not encode hubs. I made that point earlier, and here's the analysis that highlights that conclusion. 25,000 human genes, 1,565 essential genes, 1,777 which are disease genes. And as you can see, the minority of disease genes are essential genes, at least from what was in what is available to analyze in OMIM and other sources. So to try to ask the question, to try to address the question of whether or not knowing the wiring diagram might give more insight into mechanism, we need to turn to a roadmap. What's the assembly diagram? What should we use as the basis for drawing these conclusions and testing these hypotheses? Well, what we chose to use was the protein-protein interactome. And this is a real, on websites you can find lots of interactomes. Lots of people will use those to draw conclusions about pathways of disease. But many of those are imputed interactomes. These are protein-protein interactions that are physically measured in yeast-2 hybrid screens and other experiments. And it began with the first of these that Mark Vidal put together now about 10 years ago. And the argument here is that one can find clustered collections of proteins interacting with one another in this interactome, and that the essential proteins will be the hubs, as shown in red. These will largely not be responsible for complex chronic human diseases, but the disease proteins will largely be those that are in the periphery. That's the hypothesis. So, and why focus on protein-protein interactions? Why not just look at genetic networks and metabolites? Well, I would say it's important to think about the proteome because the proteome really is the physical expression of the information in the genome. The genome implies phenotype. The proteome is phenotype, or is the underpinnings of phenotype. The genome is the floor plan. The proteome is the house. And you can even say that the mature proteome is the house that's been exposed to elements in the process of weathering, the equivalent of post-translational modification. There's no way you could have predicted what this house would look like 20 years later based on the floor plan alone. And often you can't even predict how the house will appear based on the floor plan when it's newly constructed. So understanding the proteome and the protein-protein interactions within it are crucial to answering this key question about networks and information that they can provide about disease. So here we see an interactome artificially drawn. This is a simple construct for discussion at this point. And the hypothesis that Laszlo Barabasi and I raised was in this interactome, this comprehensive protein-protein interactome, can you define or identify discrete modules that contain most of the gene products that are responsible for or have been associated with a disease phenotype? Are there neighborhoods within the interactome within which there's a disease module? And we really had equipoise in framing this hypothesis. I, for one, felt it was going to be, the answer to the question would have been no. I assume that since there are many common responses to certain mechanisms of injury that span different diseases, that one would find the genes responsible for those diseases as they've been identified by conventional experimentation or GWAS to be scattered across the interactome. But I was wrong, as I'll show you. So in a paper we published last year, we explored this question in detail. we took as the diseases of interest 299 from the mesh ontology, and we picked those 299 because there were at least 20 genes or gene products that had been associated with them in the literature, or through unbiased gene expression studies, or through GWAS. Those led to our identifying over 2,400 disease-associated proteins, and recognizing, of course, that the interactome as we have built it over time, beginning with Vidal, the interactome probably only accounts for about 20% of at least our expected or predicted total number of protein-protein interactions, so it's incomplete. And then Dina Gassian, who works in Barabasi's group with Menk, Jörg Menk, developed a novel method for detecting disease modules that increased the size of the modules by one interaction distance to another protein binding partner. And that really did enrich the modules considerably and allowed us to identify linkages we otherwise wouldn't have seen. So now in the background here you see the real interactome that we used. It had at this time 13,470 nodes, over 140,000 edges. It's a fuzzball, as people in the network science realm call these. And what I've mapped here, sketched in cartoon form, are 3 disease modules as they are placed in this comprehensive interactome, one for peroxisomal disorders, one for RA, and one for MS. And notice that the RA and MS diseases have significant overlap, as you might expect, given their autoimmune underpinnings, whereas peroxisomal disorders have no overlap, essentially. And there are also a few scattered gene products that sit quite remote from the clustered modules, and this probably in part reflects the fact that the interactome is incomplete at this point in time. So what we found when we explored this, the interactome, was a couple of interesting features. One was that the smaller the module, the more compact and discrete the number of pathways was that influenced the disease phenotype. The second was that if you looked at diseases that overlapped, you could find pathways that were common to them, to get to David's comment at the introduction. And here's an example. Here we see an immune network for IgA production that sits at the interface between asthma and celiac disease. Now, this may seem obvious in retrospect. Most things seem obvious in retrospect. No one had actually shown these specific gene products as important to both diseases previously, but it was quite apparent from the analysis. So interactome-based network analysis gives insight into disease commonalities, common underlying mechanisms of disease. In addition, interactome analysis of disease networks gives insight into novel mechanisms that can serve as targets for therapy. And here's an example from the work of Vicky Parikh and Steve Chan when he was in our group. And it involves the construction of an interactome-derived pulmonary arterial hypertension network. And without going into the detail, which you can see in the paper if you're interested, notice first that it's clustered, that pretty much all the known pathways are included that have been recognized previously as associated with PAH, that there are a host of orphan gene products that haven't yet been brought into the network because the network is incomplete. But analyzing this network in an overlying microRNA network, what Vicky and Steve were able to identify was a novel microRNA 21 mechanism that serves an integrating function to stimulate Rho B and Rho kinase activity, as well as inhibit expression of BMPR2, which may be a mechanism by which non-heritable forms of PAH are accompanied by reduced BMPR2 signaling. So a novel key central mechanism for disease that was borne out in animal models in the paper as well as cell culture studies. What else can network analysis do for our understanding of disease? So here's an example of how network analysis can inform GWAS. Here we have, in a very direct way, here we have type 2 diabetes. There have been about 200 genes, perhaps a little bit more now, that have been associated with, through GWAS, type 2 diabetes phenotypes. Most of the action occurs in the first 15 to 20 genes. The rest have very tiny effect sizes, and here's a by GWAS-wide p-value. Now, if you map those genes beyond the first 15 to 18 to the interactome and follow the z-score, and the z-score here reflects the clustering of genes within the interactome as these variants were mapped to them, what you find is a z-score which begins quite high, highly statistically significant for what we call the largest connected component, or the cluster size. It starts at 9, it never goes below about 2.5, and notice these upticks which occur that are quite striking in the z-score as gene products are added one by one. And here you can see a significant uptick when column 2 was added, similarly with the addition of PRKC alpha. And just to give you another depiction of this addition, you can see that with these upticks at step 5, 7, and 12, there were significant and dramatic increases in the clustering coefficients, an expression of the degree to which these genes are brought together by the addition of one gene product. These clustering coefficients, there was an increase in their size with these simple one-step incremental additions of genes that had been linked by GWAS to the disease. And it highlighted some novel steps in regulatory pathways for calmodulin 2 signaling and PRKC alpha signaling that hadn't been appreciated previously. So network analysis also gives information then about complex signaling pathways that can underlie complex phenotypes. But let's now turn to a discussion of phenotype for the next part of the talk, because obviously, without understanding the phenotype in more detail, the molecular network analysis can ultimately prove as ineffective in exploring true causes of disease from which precision therapies can be developed as conventional GWAS is. And I turn your attention to National Academy's publication in 2011 that emphasized what was needed for our movement toward precision medicine. And just to define the universe of precision medicine for you here, I show you this slide, which illustrates in the upper row all of the omic elements of precision medicine from the genome through to the phenome, underlying regulatory features such as epigenomic features or transcriptomic regulatory features or metagenomic features linked to other genomes like the microbiome, functional genomics and proteomics and their consequences for the phenotype. And then underlying all of these elements, of course, are environmental perturbations, which can randomly often affect any one of the steps. So before we think about how to improve precision disease classification, let's remind ourselves of what the problem is with current disease classification. Diseases, as we know them now, really stem from the pathoanatomic correlations of Malpighi and Osler. Right? And remember that Osler's anatomic explorations of disease, and I apologize to all the Oslerites in the audience who may have spent time at Hopkins, that Osler's correlations of pathology to disease were all based on autopsy studies. So these were end-stage expressions of disease, and certainly weren't at all appropriate for considering early-stage or preclinical disease. That, of course, has its own limitations, but it's perpetuated this artificial distinction of diseases as focused specifically on one organ. And that's true by virtue of the structures of departments of medicine, which are organ-based. It's true by virtue of the construction and of the organizational structure of the NIH, which is largely disease organ-based. And as we learn more and more, both through network studies as well as other large data studies about the commonalities that underpin many diseases, including cancers most recently, we appreciate that these classifications are artificial and probably misinform us more than they help us. And let me just highlight for you again how one might think about networks that interconnect or conspire to create the pathophenotype. Here we have one way to look at this process. We have the primary disease genome, the secondary disease genome, and I'll give you examples of these in a moment, environmental factors that influence gene expression, the determinants of intermediate pathophenotype, such as inflammation, thrombosis, fibrosis. These are endophenotypes that are common to all diseases. All of these networks conspire to create pathophysiologic states that correlate directly to the pathophenome. So we'll go to pulmonary arterial hypertension as an example of how to classify disease in this way. Here we have, I think this is pretty up to date, primary molecular abnormalities that have been shown to be associated with PAH. The last two are fairly uncommon, or much less common than the first 4 or 5 are. We have a host of disease-modifying genes which themselves don't sustain disease expression, but would in the context of a primary molecular abnormality or other exposures. and they range from serotonin signaling down to endothelin or cerebellin 2. We have your own genetic makeup that governs intermediate pathophenotypes like thrombosis or vasospasm or inflammation. And then there are exposures, many of which are unique to PAH as risk factors such as anorexigen exposure or crotalaria toxin exposure. And these interact in complex ways in each person with PAH to produce one of several pathophenotypes, 3 of which are shown here. So this is an example of really a convergent cardiovascular phenotype, that is pulmonary arterial hypertension as measured by an increase in mean pulmonary arterial pressures. There are many other examples of convergent cardiovascular phenotypes. Here we have a hypertrophic heart. There are many causes for this kind of picture if you didn't have available to you modern molecular techniques. Those include over 1,400 variants and 11 different mutations in sarcomeric proteins causing HCM, hypertensive heart disease that's untreated hypertension, long-standing aortic stenosis, infiltrative diseases like Fabry's or Pompe's disease. They can all produce hearts that look not dissimilar to this heart. Another example is thrombotic microangiopathy, and here's an example of that, thrombosis in situ in a small vessel in the kidney. And this histopathology is the same for TTP as it is for HUS, as it is for malignant hypertension, scleroderma renal crisis, preeclampsia, HELP syndrome, antiphospholipid syndrome. So you can see just from this very straightforward collection of diseases that you all know a reasonable amount about and their histopathological expression that one can be misled if you only focused on the pathology. Another way to look at this problem begins with sickle cell anemia. And this is, if you would believe it, the classic Mendelian phenotype, one mutation in the 6th position of the beta chain of hemoglobin, causing sickle hemoglobin. But there are many disease-modifying genes, some of which are positive, some of which are negative. Again, the determinants of intermediate pathophenotype, different exposures like hypoxia, dehydration, infection, and a host of different pathophenotypes, from hemolytic anemia to stroke to bony infarction and so forth. So this is an example of a divergent set of cardiovascular phenotypes or phenotypes. And others include aortic stenosis, which can present with syncope or heart failure or angina or some combination, or antiphospholipid syndrome, which can present with venous thromboembolism or thrombotic stroke or coronary thrombosis or liver reticularis. So you have to be aware of these differences in convergence and divergence as you challenge disease classification. And I want to emphasize, return now to the convergent phenotypes because there are more of these. Many distinct diseases have common phenotypes. And a second point is that common treatments, disease co-occurrence in the same person at a frequency greater than the play of chance, and GWAS, as well as network analyses of the sort I showed you earlier, really suggest that there are common underlying mechanisms for many distinct, so-called distinct and different diseases. Now here's I don't expect that you can see anything on the panel, but it's taken from a paper by Rajetsky in PNAS from about a few years ago from his work in which he asked the question, looking at 1.5 million data points in the Medicare database, do diseases co-occur in the same person at a frequency greater than the play of chance? And he's not talking about diseases that may lead from one to another, such as COPD and squamous cell carcinoma of the lung. But he was looking at other diseases that were not known to have any relationship mechanistically or from an exposure perspective previously. And he found that that was, in fact, the case, that about 20% of patients had diseases that occurred, had more than two or more diseases that occurred greater than the play of chance found in this population. I already showed you a Manhattan plot from the Wellcome Trust Case Control Consortium to talk about linear thinking. But this is the full panel from that study, which highlights, again, different GWAS loci, the same GWAS locus or loci, common to different diseases. And I put in purple ovals a few of those, including 2 that link Crohn's and type 2 diabetes, and 3 that link RA and type 1 diabetes. So how would you change the taxonomy for disease? What needs to be considered? Well, I take this quote from the monograph that I showed you the cover of a few slides ago. in which they point out that a new disease taxonomy should be developed that would describe and define diseases based on their intrinsic biology, in addition to traditional physical signs and symptoms, go beyond description and be directly linked to mechanism, and be highly dynamic, continuously incorporating new emerging information. Makes good sense. Huh? So how can we improve clinical phenotyping? What should we do? Should we simply continue to finely dice rather than be a lumper? All different subgroups of patients with heart failure, or all different subgroups of patients with venous thromboembolic disease. Well, you can do that, but it should be based on rational, objective measures that distinguish the subcategories. We should define new unbiased phenotypes, and these can be garnered from existing data sets or new data sets. And some of the information that's gathered to put these data sets together should be orthogonal information. That is, there should be no bias that leads you only to gather information on cardiovascular system function if you're looking at heart failure. There's no reason to believe, in fact, there's every reason to disbelieve the fact that the pathways that are active in heart failure only involve the heart or vasculature. And in fact, these so-called orthogonal phenotypic features may allow you to subcategorize patients a little more definitively. And you need to build the analytic and cognitive base for integration and interpretation. I list this as the last point. It's actually the hardest point. So there are ongoing approaches to gathering this information and creating exquisite phenotypes, as they're called. There's dbGaP, which is a database of genotypes and phenotypes that the NIH has put together. Lots of data, hard to wade through it, different formats. It's improving. But there's been no rhyme or reason as to what should go into the database aside from genotypic and phenotypic information. So putting together a more rigorous infrastructure for the kinds of data entry so that they can be analyzed with some kind of consistency would be crucial, and dbGaP doesn't do that. PheWAS is another strategy. This was developed by Denny and colleagues, and it's the phenotype antithesis to GWAS. Which is to say that you take the 750-odd defined phenotypes that exist in medical databases, and you basically sweep through them looking for links to SNPs. And fortunately, in this paper, they showed that there was a correlation between positive GWAS and positive PheWAS, but there were also differences. So you can gather more information in this way. Of course, you're limited by the challenges of these phenotypes as they're currently defined. Electronic medical record-based analysis, this is all the rage. I know it's going on here as well as other places. I think this is clearly an important effort, but it also needs to be standardized in some way and be as unbiased as possible and as inclusive as possible. And one could also repurpose existing clinical trial data sets or drug trials, using drug trials as systems perturbations if you have time sequence data in gene expression or in phenotype changes. Obviously, time sequence changes are extraordinarily important in trying to attribute causation or benefit to gene products or treatments, respectively. This slide was courtesy of Callum McRae, who highlighted a couple of elements of novel orthogonal phenotypes as facial recognition. You can imagine the UK Biobank actually has been gathering data in this way, using as the following example to make that point. Patients enter their clinical data in front of their computers, send in data to their primary care physicians about their weight and about their blood pressure and pulse if they're being treated for heart failure. And as they sit in front of the computer, the cadence of their typing is quantified, measured in some way. Spontaneous blink rates and facial expressions are monitored, as are pupillary signs of various sorts. And skewed vision, pupillary size in view of the light that exists around the computer screen. These are all unrelated to the disease for which the patient's being treated, but gather useful information, some of which have been shown to be associated for reasons that are not clear to certain outcomes or certain phenotypes. There's also now an increasing interest by Google and other groups to develop these flexible skin sensors, one of which can assess over 60 different parameters non-invasively and continuously, in which the relationships to disease are completely unknown. Those could include skin traits like cell-cell communication and the links to cardiac diseases. Naxos disease is one such disease in which you might expect there to be some commonality here. Salt secretion in pulmonary or intestinal diseases like CF. Fluid content of cells, their consequences for skin impedance and renal or cardiac diseases like renal failure and heart failure, respectively. So we're only limited by our imagination and technology, and people like all of us here in this audience today, being in academic medical centers, aren't limited by imagination. It's really more technology. And I think many of the big technology companies are quite interested in this area, including Google and Amazon and others. So more to come. So let's spend the last few minutes talking about therapeutics and how network medicine can inform therapeutics more rigorously and rationally. So here we have, up through 2013, the FDA-approved drugs per year. And as you can see, they've decayed since the mid-90s. A couple of blips here, but by and large, they've been in the 20 to 30 range per year. And if you ask anyone from the pharma industry why there is declining productivity or success in approved drugs, they'll give you at least 4 of these answers. They'll of course, emphasize the regulatory environment. They'll talk about the increasing need to explore novel targets, but they're better at that now than they ever were with genomic information, so that's a hollow argument. Easy targets have been exhausted. Well, that's certainly true. And there's an increasing attrition rate for developing drugs, in part because of the regulatory environment, and that's true as well. But I would say the greatest, or one of the greatest reasons for declining productivity has to do with a flawed reductionist approach to drug development. The constant search for identifying a single drug target and drugging it with a single magic bullet. Even the FDA insists upon this as a means of getting approval. So let's just turn to our disease module construct here and explore why that is a problem. Now here we have a drug target, and we can learn everything we need to know about it from structural biological techniques that I'll review in the next slide. And we screen those structured molecules against large virtual drug libraries and find those that might likely bind to the important regulatory sites, theoretically, and then test them in real screens. And that's how drugs are discovered. And this target-based screening has been facilitated by the genomic data sets that give us a greater pool of targets that can be explored, and of course, great structural tools that have been improving rapidly over the last many years, including X-ray crystallography of proteins, NMR spectroscopy, computational modeling, newer strategies for electron microscopy at low temperatures. And then there are these large real and virtual compound libraries. Many big pharma companies have compound libraries that range from 1 to 2 million or more. And high throughput screening technologies have also become certainly readily mechanized through robotics. And yet with all that, if you look at the percentage of new medical entities that have been identified by phenotype screening with all these powerful techniques versus target-based screening, as Swinney and Anthony did, and this was over the preceding, I think, 2 or 3 years, what they found was that there were almost twice as many drugs identified by phenotype-based screening compared to target-based screening. That's not true for follower drugs. As you expect, in the case of follower drugs, you simply modify a member of a drug class and show that it has the same effect. So functional phenotype screening makes sense from a network perspective because you're now exploring the drug target's function in its appropriate functional context. And that is likely to give you much more useful information. And if you don't do it, you can get in trouble. And I'll show you an example of the trouble you can get into here. This is a panel taken from a paper by Nick Wagley, who's an oncologist at the Farber, one of my former chief residents. And it's a study. It's one of the first case reports of the development of resistance to vemurafimab, a BRAF mutant BRAF inhibitor. So here's a patient. He was a young man, late 30s, who had widely metastatic melanoma, lots of subcutaneous nodules. The BRAF mutation was identified with the biopsy of one of the nodules. It's found in about 80% of melanomas. He was given vemurafimab, the first of the BRAF mutant inhibitors. 15 weeks later, he looks great. The red light even shows how robust he looks. And all of his subcutaneous nodules have disappeared virtually. But then we move forward another 8 weeks or so, and the tumor comes back with a vengeance. And now when it's biopsied, there's a mutation downstream in this important oncogenic pathway at MEK1. Now, it's clear from the work of Vogelstein and others, there probably aren't more than about 14 or 15 key oncogenic pathways, the balance of which governs oncogenesis in virtually all solid tumors. And this is one of them. And if you know that resistance develops or can develop in this pathway at one of two sites, then why not drug the entire pathway from the start? That's in fact what was done in a clinical study that was published at the end of 2012, in which a BRAF inhibitor and a MEK inhibitor were used in combination and showed improved outcome. And I would say that this is a reason to think about so-called rational polypharmacy, understanding the pathways that govern a disease or govern a phenotype that you want to restore the health of, and then using a variety of computational techniques to determine how best to use combined therapies that optimize benefit and minimize toxicity. And this is an example of using this combinatorial approach theoretically and then experimentally that Garmaruti and Handy did in our lab, looking at the pathway that governs nitric oxide production, identifying three key steps in this complex set of reactions that optimally govern optimal NO output. And that was confirmed in our cell-based studies. Another therapeutic story to tell you about that highlights the benefit of network analysis comes from the work of Amitav Sharma and Scott Weiss and Ed Silverman, in which they took the asthma module, rather dense. Here you see the C genes that were derived from the literature in GWAS that are linked to asthma, and then the diamond genes, those that are interspersed among them from this next neighbor analysis that I mentioned earlier, creating this large module. And by exploring this module, what they were able to uncover is a novel mechanism for resistance to corticosteroids in asthma, and it has to do with a novel pathway that corticosteroids can induce in patients with the right variants that stimulate NF-kappa B activation, which is an inflammatory transcription factor, as you know, an important signaling pathway. So that if you, as a result, if you could identify this signature in a patient, you would perhaps be more resistant to beginning with corticosteroid therapy when inflammation is exacerbated. In another recent bit of work that involves a network analysis of drug therapies, Rusheng Wang and our group used network approaches to repurpose drugs and identify adverse drug reactions using the cardiovascular drug targets that existed in the literature, mapping them to the interactome, and then linking them to other targets for which drugs have been developed, but not for cardiovascular purposes, that are associated with them through the interactome. So she created a bipartite graph, and as a result of this analysis, we were able to identify some novel drugs, which are shown not in circles here, but linked to the targets, that can adversely or beneficially affect pathways that influence myocardial infarction in this case. So the last story has to do with the placebo and how you can use a network approach to understand placebo effects. This work isn't published yet, so it's incomplete, but it's interesting, I think. So the placebo effect is real. I think we'll all agree to that. And as it turns out, there have been several GWAS studies that have identified some loci, which ultimately led to the identification of 23 genes that appear to link to a positive placebo effect. Now, you can imagine that how you define a placebo effect will differ study to study and disease to disease. And I realize that there are all kinds of flaws in the logic of this approach, but it's given us at least an infrastructure to begin to work on the question a little more rigorously. So I asked Rusheng Wang and Catherine Hall to address this question. Do these placebo seed genes comprise a module in the interactome? Are they discretely localized? Do they construct or identify or comprise a placebome? And the answer is yes. And this hypothesis, I should say, was based on a work that Catherine Hall did, who's a terrific young member of our faculty, an epidemiologist, statistician, who had explored the women's health study for SNPs that linked to a positive placebo response in the arm of women who were given aspirin or placebo for for primary prevention. And what she found was a very prevalent, to the tune of 47%, variant of catecholomethyltransferase, which conferred protection against events, even in the absence of aspirin. Aspirin provided no additional benefit. And it probably has to do with the fact that this heightened, this COMT variant of heightened activity decreased circulating catecholamines and thereby increased platelet activation threshold. At least that's the working hypothesis. So based on that, I asked her to look at other GWAS studies of placebo effects. We developed this pooled these 23C genes together and built this placebome, and now are using it to answer the following question. Do these genes, does the proximity of the placebome being discrete in the interactome to a disease module, does that proximity define the likelihood that there'll be a greater placebo effect in treatment for that disease as compared to diseases that are remote from it? And that is ongoing work. But think about what the other implications are if we do find and verify a set of pathways that govern the placebo response that are linked. You can imagine that you might do molecular analyses of some sort, either looking for SNPs or looking for some other signature, that identifies a person you're about to enroll in a clinical trial as being more or less susceptible to placebo effects. By eliminating the placebo-enriched population, you would decrease the sample size necessary to see an effect. You can also think, and I'm sure drug companies would love this, that you might want to drug the placebo for certain diseases. Rather than give somebody with low back pain chronic opioids, there might be ways to activate certain subcomponents of this placebo set of pathways that would provide the same benefit. So the last couple of slides just highlight where we are with network medicine as it links to precision medicine and personalized medicine. So clearly, data acquisition platforms are essential. Genomic and phenomic components are crucial elements in their broadest sense, the most inclusive sense. And the analytics, which we didn't really spend a lot of time talking about except for some of the features of the network analyses, are really going to be where all the money is, because that's going to lead to appropriate medical decision analysis that will influence patient outcome. So we really see research continuum, and this slide was provided kindly by Elliot Antman, two different, if you will, profiles of discovery science versus population science with clinical trials sitting in between in the era of precision medicine. In the case of discovery science, the play of chance is profound, and bias and confounding are less so. In the case of population science, bias and confounding are profound, and the play of chance is less so. With clinical trials, they must be optimized, of course, for internal validity and external validity, given the clash, if you will, of the shortcomings of these two strategies for disease determinant detection and treatment. Okay. So the last slide here, just which is taken from two papers, one by Hawgood in Science Translational Medicine, another by Jess Mega in JAMA, that highlights where we are, highlights as well the fact that I've talked about genomics, mentioned metagenomics and exposures, didn't talk about behaviors, which are at least as important as these other networks. These all create networks of networks, which is in itself a novel science. There's a very nice paper on obesity networks in the New England Journal from about 3 years ago with an editorial by Barabasi that highlights these points. Clearly, even if you have, whether or not you have genomic or environmental predispositions to obesity, if you hang around in your social circle with chubby people, you're more likely to eat with impunity, which only can make your problem worse. That's a simple analogy, but there are many like it for other diseases. So we're moving from this era of population medicine. Personalized medicine was the ideal, but is not practical for a whole host of reasons that we could talk about in the discussion. Precision medicine, where we refine subphenotypes of an inclusive population that optimize the chance for seeing great effects and minimal toxicities with minimal number and minimal costs of participants. I'll thank the people who did the work I mentioned from our group, and also end with this discussion question slide for house staff, which David asked me to put in the talk, but I'm happy to answer any questions you have at all. Thank you for your attention.
Dr. Loscalzo, thank you very much. That was fantastic. We probably have some questions from the house staff and students. Let's take those first.
We actually had a question that was sent over from another site where this is being streamed, which is, what do you think the impact of network medicine will be on medical education? Because it seems that it kind of defies all of the systems-based learning that we're doing in the first 2 years and seems to complicate that significantly.
Well, I'll mention two initiatives that we have that will begin to answer the question. One is that we have a division of network medicine in the Department of Medicine. So we're using that as the basis for creating the analyses for each of these diseases, examples of which I gave you here. And that will be a site in which people will train, medical students, postdocs, fellows. So education is one element of the answer to the question. The second is that there's actually a textbook of network medicine which will be published later this year that we put together that will, I think, begin to define the discipline a little more objectively and a little more clearly. And then the third is that I think it does complicate the way we teach medicine now from a case-based perspective, but not entirely. I think once you compare those cases to what the students actually see on the wards, they'll notice that there are very few patients who mimic exactly what they saw in those cases on the wards. And that's because of these complex, individualized nuances that exist that are contributed to by their networked relationships.
So I think the House staff would be really interested in the part of your talk that you said was almost impossible, the step to take, which is personalized medicine.
Oh, yeah.
So tell us what you conceive of as the challenges moving from precision medicine to personalized medicine. And if you projected 5 to 10 years forward, do you see us getting there?
Yeah. Well, the major challenge is just the cost of the enterprise and the degree to which each person would have to be so finely diced in terms of their molecular infrastructure to idealize a therapy just for him or her. And I say I don't distinguish very much precision from personalized medicine. I think precision medicine is rational and practical personalized medicine because you have a big enough cohort of people who have common enough features, either molecularly or clinically, that you can feel confident that that is the, if you will, the lowest common denominator to which you can bring the entire population and still justify the cost that would be involved for developing therapeutic strategies that are specific to them.
And you'd say that network medicine clearly identifies the dynamic nature of the diseases that we take care of, and in that sense, different treatments at different phases of the disease.
Yes. And I didn't really have a chance to talk very much about time sequence data except for that one comment, but yeah, I mean, questions like, do the networks get rewired over time? In the case of a disease like BCR-ABL diseases, CML, you can imagine you're creating a new protein. It has new protein binding partners. That's an obvious one. But what are the consequences of a variant in a gene product with respect to its binding affinities for a binding partner? And then what does that do to the dynamics of that network? Each of those steps needs to be explored individually to know if there's a powerful effect or not that's worth pursuing treatment for.
Other questions? Yeah, Bill.
Question is sort of philosophical one. Essentially what you're proposing is that by network analyses and precision medicine, you can identify a more rational molecular basis to treat disease. But how do you actually test that? In other words, you're essentially generating thousands of subgroups. And so if you want to develop a new cardiovascular drug and you hypothesize a 15% relative risk reduction in a particular population, you might need 10,000 patients to show that. But now if we're interested in all the subgroups that may come out of that, were you ever going to define risk and benefit in a particular subgroup with any economy that makes sense?
Yeah. So I think we have to take a cue from the oncology world because they've been doing this semi-empirically for a number of years, right? So they really perfected the notion of adaptive trial design. They always suffer from limited sample sizes. They don't deal with the 10,000 patients we do, and yet they get drugs approved, and they help people in most cases. So if you can define, the argument here is that if you define just the right size cohort, and I can't tell you what that size is because that requires an economic analysis. Let's say it's 10 or 100 or 1,000 patients instead of 10,000 patients. And you know that by restricting the definition to be as exclusive as possible, that you will optimize the chance for a beneficial effect, then the effect size will be greater and the sample size needed will be less. I mean, that's the thinking, the logic behind it.
But there was one other question, yeah. Let's take this as the last question.
All right, thank you. Amazing presentation, really enjoyed it. So I apologize, I am a little bit of a science nerd, so I'm going to ask kind of a weird question. So seeing now that we're looking now at the micro scale, looking at genomics, looking at the microbiome, then we're, say, looking at interactions from person to person. I imagine at a point we're going to start looking at levels in the community, like a geographical or even political, possibly, just a bigger climate, to say. My question is, what do you think medicine of the future is going to look like? You're on the vanguard, I believe, of what medicine is now approaching, and there are financial constraints and technological constraints as well. But I think over time, seeing how technology has progressed, we'll probably reach a point where it would be feasible to have all this data in real time while being in the patient's room, in theory. My question is, if you had to just sit and think, what would the physician of 2100 look like? What would that be?
It's a wonderful question, because it's sort of worth probably 3 hours of discussion at a symposium. But I'll give you, perhaps the best way to answer it is to at least share with you what I think are things that are invariant about the way we as physicians should approach patients, and then sort of backtrack to the nerdier part of your question. So, with all due respect, so I think the first thing is that with the electronic medical record and the imposition of all the stresses that's caused for most of us these days, the patients are getting physically more and more removed from patients. I mean, all the young faculty at our place will, as I'm sure most of you do, type and look over the computer to talk to their patients. Now, I'm old-fashioned enough, and I have a more constrained outpatient clinic volume that I have the luxury of not having to do that, and I've insisted that I won't, because eye-to-eye contact, physical contact, those features of the physical examination, even if they're not predictively accurate in all cases, are absolutely essential for creating the right kind of bond as a physician. And I think we should never lose sight of that, no matter how precise we think we can get about predicting who's going to die from what or who needs what intervention. So that's the first point. Second point is that as we gather this information, how we convey it to the patient, especially in an era, in an evolving era in which patients are more and more involved in their own decisions, in decision-making about their own care, is going to be incredibly important. And how you distill these very complicated ideas down to something that a bright patient, but not versed in biology or science, can understand is going to be a critical element that needs to evolve in tandem with the technology. And the third thing I'd say is that while this may seem daunting in terms of the levels of complexity, and you outlined them nicely, we talked about social networks and how they influence behavior and how that could influence disease. And I think you're right. Political persuasion might do the same, both as a consequence and as a cause. I think understanding those kinds of interactions, those sociological determinants in more detail will be important. Not to say that you can intervene, but at least you can alert the patient to the fact that having the following behavior or participating in the following activities has been associated with. The one last point I make is that while these I began to make this point but lost my way there. While these networks seem incredibly daunting in their complexity, one of the critical features of the people who analyze networks now is to try to reduce them to their bare minimum. There's a lot of overdetermined information here in these networks, just by the way they evolved. What's the bare minimal amount of information you need to know in the asthma network to predict precisely how someone develops asthma under what circumstances and how you can treat it? And they can be reduced. That will take a lot of trial and error to do them.